Okay, so um, organogenesis. I think you have some sort of rough idea, no? Remember when I told you about totipotency, where any cell or any tissue of a plant, let's say you took a xylem cell or a phloem cell or a cortex cell and you regrew into a whole plant, no? Then totipotency is similar to that, okay? But here we are going to regenerate an organ, okay? If I say organ, okay, suppose this is me, this is an organism, right? What are my organs? My hand is an organ, okay? My legs, my toes, my mouth, my lips, okay, these are all organs. What about my lungs? Yes, my lungs are an organ. Then my liver is an organ. My intestines are organs. What else? My heart is an organ. Then what about my skin? Is it an organ? My skin. Well, your GK, the skin is actually the largest organ of a human. Okay, your entire skin. I'm not talking about one cell, one skin cell. No, but the entire organ of your skin. Okay, the entire skin is an organ. So I think you understand, no? My hand is an organ, my arm is an organ, my leg, my knee, all these are organs, okay? Then what about plants? This flower is an organ, yes. The stem is an organ. The leaf is an organ. The shoot, let's say if this is suppose a shoot, a shoot is an organ. Then the roots are an organs. Okay, so not that you will be cutting and observing the cells under the microscope. No, this is a cell. Okay, just try to remember the levels of organization that you did in ecology last year. No, you started with a cell, no, plant cell. Then you went to tissue. Okay, suppose these are your xylem tissues. Then a group of xylem tissues. Then it gives you the from cell to tissue. Then many tissues together, the xylem, the phloem, it gave you the vascular bundle. This is an organ, okay? Or suppose I start with a plant cell, then a group of leaf cells, okay? These group of leaf cells are tissues. Then your organ will be leaf, okay? Then after organ comes organism is your plant, okay? So this is your organism. So I think you understand the levels of organization, no? So if you talk about the organogenesis, means we are going to regenerate a plant, a whole plant from the organs, okay? Organ like the root, shoot, buds, etc. I'll make it bigger. It's too big now, no? Usually, here, this sentence, no? This one. Usually, even outside the lab, okay? Organogenesis we are doing in the lab condition. But usually, in nature, no? Totipotency, it happens, okay? For example, this is a sunflower plant, no? This is a sunflower plant, okay? Then, what you do is you cut this one, okay? No. Think about a rose plant, okay? This is a rose plant. I don't know how to draw a rose. Then you cut it. Then what happens? It still grows, and then it produces like a bush, like a rose bush, no? This is the rose bush, okay? It starts to bud advantageously. Okay, instead of just growing upwards, it becomes into a rose bush, no? Because at the tip of this flower, this bud, no, let's say this is where the flower bud will, at the tip, right, there are the meristematic cells. Remember, I've been telling you these cells that are always reproducing, right? These meristematic cells are always alive. So whether you cut the head of a rose, it will still bloom, it will still grow. Okay, plants will still grow, branches will still grow. Similarly, in the root, okay, sometimes you see, you know, these Japanese people, they used to trim the roots of their trees and all. That is so that the roots will branch out. Okay, they will become adventitious roots. So here we can create a complete plan where there is no meristematic cells. Okay, because the meristematic cells are only present in the shoot or in the root tip. Okay, shoot tip or root tip. The new organs are formed in two stages. In the first phase, the cells, they divide and form undifferentiated cells. Okay, remember the callus, I have been telling you, it's those cells that await the orders. Okay, you can become a shoot, you can become a stem, you can become a root, so they will form the callus. Then in the second phase, they will differentiate, means that callus in the second phase already got its own different activity. This one becomes root, then this one becomes shoot, this one becomes stem. It already got its direction. So the factors that induce or influence age, as I told you, we should take young meristematic organs, no, young organs. No point collecting old plant parts. Okay, they will become weaker with age. Then the ploidy level. Here, what they have written is after organogenesis, different ploidy level is noticed in various plants. Okay, means they, instead of being diploid or tetraploid, they have maybe a different set of cells. Then plant hormones, they also affect the organogenesis. As I told you, if you add auxin, it will induce root formation. If you add cytokinin, it will induce shoot. Okay, then for another chemical, phosphate concentration, it will favor shoot formation. Okay, so these are the factors. Then photoperiodism and vernalization, I think we have done last year. No, I think so. So depending on, we can induce these organogenesis by inducing photoperiodism and vernalization. Okay, applying cold treatment and light treatment. So they have performed these experiments and they saw that you could induce these organogenesis. Okay, on a plant organ which will give rise to a complete plant. Okay, so I told you there were two methods, no? The first method is directly from the explant. Okay, you can produce the organs directly from explant. Explant means any part of a plant which you are cutting. Okay, like here, this person has cut the bud, then another person cut the shoot, another person cut the leaf, and any part you cut, you can regenerate the whole plant. Okay, let's look at the diagram. I think I've explained this diagram in that production of haploids or something, no? Okay, we'll just revise. So they took an explant, right? Any part of your plant is, 1 minute. So any part of your plant you cut, that is your explant. So here in this diagram, these are the examples of explants. Then the shoot had formed. Okay, here because the apical meristem is present, no, directly it'll become ill form. Then here they are culturing the node. Okay, means they put it on the media. Okay, here, how should I say? What I mean is, for the shoot and the stem, you can culture them on solid media, and for here, the rest, you can culture them on the liquid medium. Let me just tell you what it means. Suppose you have cut the small tissues, small cells, no? You cut one cell, okay? You cut one cell. You cannot take a cell and place it in a Petri plate on a solid media because the cell is so tiny, no? So what you do is, in a this is a conical flask, no? In a broth, okay? Broth means yes. If I give you a complan to drink, you will add it in water, no? So that is a nutritive media, and you drank it, okay? Then if I gave you that complan and I tell you add the agar, it will become semi-solid. So you will eat that complan like a jelly. So that is your semi-solid media. But a liquid media, here I have not added agar, okay? So it is liquid. So directly with the forceps or with the test tube, so difficult to draw with my finger. So directly with the forceps, suppose, or with a needle, they have extracted one cell, and then you just dip in this broth. So simply the cells will float here in this broth, no? And then all the nutrition is there, the hormones are there, so it will induce that callus to form. And then depending on what you want, a shoot or a root, you can induce. So I think here this part is clear, I think. This one, why we are creating callus, means the cells to form inside the broth, okay, or the suspension culture, okay, because they are so tiny. Then here the organs have formed, no? And then here somatic embryogenesis has formed. Remember when you dissected the cucumber? This is your cucumber seed you took, and then you dissected, and then you got that, what was that? Hostoria, no? And then some of you here, you got a heart-shaped embryo. You got a heart-shaped embryo. So similarly here, you can you can create a embryo artificially, okay? You don't have to open up the seed, then find that hostoria, then find the what was this called? I forgot. I forgot. Ugh. Then locate the embryo inside this. What is this called? Dissection of embryo. What is it? It was the endosperm. I had forgotten. You dissected the endosperm, no? And then inside the endosperm, this was the host, this is the endosperm, right? The cellular endosperm. Then inside some of you got the heart-shaped embryo. So in this method, you don't need to wait for the plant to produce a seed, then then dissect, then see inside. Here directly you can get your somatic embryos. Somatic embryos means the embryo that is formed without fertilization. Okay, somatic cells are the vegetative cell. I mean, somatic cells are not the vegetative cell. They are not the germ cell. Okay, like for me, my somatic cell is any part of my body other than my ova. You get it? Other than my eggs, any part is the diploid part is your somatic. So same in plants, any root, shoot, stem, except for pollen grain and, yes, you are right, and the ovule, okay, those are the vegetative cell, and the rest are the somatic embryos, okay, if you create embryo from there. So here they have grown the callus, the cells here, and then the somatic embryos were produced, means the artificial embryo, and then you can transfer it to a rooting media, okay, means an agar that has auxins for roots, and then cytokinin for shoot, and then When the regenerated plant is ready, it's strong enough, you can transfer to the soil and then to the nursery and then to the field. Okay.